Good afternoon, everyone. Today, we embark on a journey to demystify the intricacy of past year exam question for March 2022 for A-level students. Examining the past offers us a compass to navigate the challenges ahead, empowering us with the insight and strategies to conquer the present. Let's delve into a treasure trove of past exam, unlocking the key to success and mastering the art of academic excellence. Question number 1. A student used a stage micrometer scale to calibrate an eyepiece reticule. So the diagram shows the view of both the stage micrometer scale and the eyepiece reticule seen by the student. The divisions on the stage micrometer scales are 0.1 millimeter apart. The student removed the stage micrometer scale and viewed a slide with the blood cells on it. And the same lenses were used so that the magnification remained unchanged. The student measured the diameter of one of the white blood cells on the side using the eyepiece reticule and recorded that it was 8 eyepiece units. What is the correct diameter of these white blood cells in micrometers? The answer will be 20. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. 1 is represent mRNA passes through to the ribosome. 2 produces the mitotic spindle during cell division. And 3 packaging of hydrolytic enzyme that will remain in the cells. The appearances were listed by letter. V represent membrane which surround and enclose inner cavity. W represent non-membrane bound spherical structures. X is a double membrane interspersed with pores, and Y represent non-membrane bound cylindrical structure, and Z is a membrane bound sac arranged as a flattened stack. Which student correctly matched the number functions with the appearance of the cell structure? The answer will be 1. mRNA passes through to the ribosome is actually referring to the appearance when a double membrane interspersed with pores. Number 2 is actually the appearance of non-membrane bound cylindrical structures. And number 3, which is packaging of hydrolytic enzyme that will remain in the cells, is actually the appearance of membrane-bound sac, which is arranged as a flattened stack. Which size range would include most prokaryotic cells? The correct options is B, where the most prokaryotic cells, such as bacteria, typically falls within this size range. What is present in a typical prokaryotic cells and a typical eukaryotic cells? The correct answer is A, which is 17 as ribosomes. Both prokaryotic and eukaryotic cells have ribosomes, but they differ in size. Prokaryotic cells have 70S ribosomes, while eukaryotic cells have 80S ribosomes. However, mitochondria and chloroplasts within eukaryotic cells also contain 70S ribosomes, which are similar to those found in prokaryotes. Which statement about viruses is correct? The correct statement about the viruses is A. They all have a capsid made of protein. A capsid is actually made of protein, and it is a fundamental component of all viruses. Enclosing their genetic material, not all viruses contain RNA, but some have DNA. Not all have an outer envelope, which is made of phospholipids. Envelope viruses do, but not envelope viruses do not. And viruses do not contain ribosomes, including ATS ribosome. Sample of glucose sucrose and a mixture of glucose and sucrose were divided into two halves, M and N. M was then tested with Benedict solution, and N was boiled with dilute hydrochloric acid, neutralized, and then tested with Benedict solution. The color of the solutions was compared to the color standard, which table identified the correct color changes for this sample. The answer for this will be, for the glucose, when they tested with Benedict solution, they will change it to yellow. And for the sucrose, it will change it to blue. And for the mixture, it will change to yellow. Meanwhile, when the glucose was boiled with dilute hydrochloric acid, neutralized, and then tested with Benedict solution, it will change color to yellow. Meanwhile, sucrose, when it was boiled with dilute hydrochloric acid, neutralized, and then tested with Benedict solution, it will change to yellow. And also, for the mixture solution, when it was boiled with dilute hydrochloric acid, neutralized, and then tested with Benedict solution, it will change to red. Number 7. Which molecule contain 1, glycosidic bond? The answer will be C. And the diagram shows these triglycerides X, Y, and Z. Which row is correct for these triglycerides? The answer given here is X, Y, and Z contain saturated fatty acids, and X and Y contain unsaturated fatty acids, and also X and Y contain more than 2 different fatty acids. Some food contain hydrogenated vegetable fats. These are unsaturated fats that have been converted to saturated fats. Which property of the fats will have changed? The correct answer given here is A. Their hydrocarbon change will fit together more closely. Hydrogenation convert unsaturated fat, which have 1 or more double bond in their hydrocarbon change, into saturated fats, which have no double bonds. And this process makes the hydrocarbon change strengthen, allowing them to fit together more closely. And this often results in the fats being solid at room temperature rather than remaining liquid. A polypeptide contains a specific number of amino acids, which is N. How many peptide bonds are present in this polypeptide? The answer is N minus 1. In a polypeptide, the number of peptide bonds is always 1 less than the number of amino acids. This is because each peptide bond links 2 amino acids together. Thus, for a polypeptide with N amino acids, there will be N minus 1. N minus 1 is a peptide bond. Which statement is correct? The correct statement is D. Deoxyribose, fructose, and ribose are all monosaccharides. Deoxyribose and ribose are both 5-carbon sugar pentoses, and fructose is a 6-carbon sugar, hexose, and all 3 are simple sugar or monosaccharides. A student used colorimetry to monitor the hydrolysis of a protein by a protease enzyme. The student used blue red solution to determine the concentration of protein in the hydrolysis reaction. The student produced a calibration curve using known concentration of protein. Which diagram shows the calibration curve? So, for this question, the answer will be B. The adsorbent versus protein concentration. And you get this value. A student completed an experiment to measure how increasing concentration of substrate affect the rate of an enzyme control reaction. The student then repeated the experiment after adding a fixed quantity of a reversible competitive inhibitor, which row described the effect of a reversible competitive inhibitors on enzyme activity. Let's look at the explanation. The correct answer is B, which is the attachment of inhibitor at active site will be yes, and the effect of increasing substrate concentration on rate of enzyme control reaction will be rate increases. This describes the effect of a reversible competitive inhibitor accurately. Reversible competitor inhibitor bind to the active sites of the enzyme, preventing the substrate from binding. However, if the concentration of the substrate is increased sufficiently, it can outcompete the inhibitor for the active site, thereby increasing the rate of the enzyme will control the reactions. Number 14, the diagram shows a liposome. Liposome can be used to move therapeutic drugs into cells of the body to treat conditions such as cancer, which row shows the property of a drug that could be transported in the section of the liposome, level 1 and 2. For the property of drug transported in location 1 is referring to hydrophobic, and property of drugs transported in location 2 is referring to hydrophilic. Some processes occurring in cells are listed. One is the endosynthesis of water into cell. Two, number one is actually, this is the process to show the use of ATP in the cells. Endosynthesis of water into cell, and this referring to this is typically a form of active transport such as phenosynthesis, which require ATP. And for statement number 2, exosynthesis of enzymes from cells is the process which involves the active transport of material out of the cell, which also require ATP. And number 4, phagocytosis of dead cells by macrophages is the process. This is a form of endosynthesis where the cells engulf large particles which require ATP. And facilitating diffusion of glucose into red blood cells, which is a process 3, does not require ATP. And as it is a form of passive transport where glucose moves down its concentration gradient through a membrane protein. So the answer which require the process use ATP is only 1, 2, and 4. Number 16, the graph shows changes in the concentration of a solute inside a cell. You have the graph here is a concentration of solute inside cell versus time. And what is actually explaining these changes in the concentration? First is actually explanation about evolution. And number 3 is exosynthesis. And number 4 is a process of osmosis. Number 17, the indicator crescent rate changes from red to yellow when put into acid. 4 blocks of agar containing crescent rate were cut to different sizes measured in millimeters. The block was submerged in acid. All other variables were kept constant. The time taken for each of the blocks to completely turn yellow was recorded. Which of the 4 blocks became completely yellow most quickly? So in this question, to determine which block became completely yellow most quickly, we need to consider the surface area to volume ratio. Meaning that the higher the surface area to volume ratio, the more efficiently the acid can diffuse into the block, and the faster it will turn yellow. Let's calculate the surface area to volume ratio for each block. Example, if you look for the answer number A, where 3 multiplied by 30 multiplied by 30, you get the volume of 2,700 millimeter per cube. And for the surface area, you get the value of 2,116 millimeter. And for the surface area to volume ratio, you get the value of 2,160 divided by 2,700, you will get the value of 0.8. Meanwhile, for the answer number B, you get the value of 1. And for the answer number C, you get the value of 0.67. And for the answer number D, you get the value of 0.5. So, the block with the highest surface area to volume ratio is B, 6 multiplied by 6 multiplied by 6, where you get the surface area to volume ratio is about 1, which therefore we consider the block B, 6 multiplied by 6 multiplied by 6 would become completely yellow most quickly. So the answer will be B. Number 18, which processes require mitosis? The correct answer will be 1, 3, and 4, which is the cloning of T lymphocyte. During T lymphocyte cloning, mitosis is required to produce identical copies of the original T lymphocyte. And the growth of multicellular organism from a single cell, as a multicellular organism grows, its cells divide to mitosis to increase in number and contribute to growth and development, which is number 3, process number 3. And the reproductions of unicellular eukaryotes is a unicellular eukaryote reproduce asexually through mitosis to produce genetically identical offspring. The answer will be 1, 3, and 4. Which event listed are part of the cell cycle? So the events that are part of the cell cycles are interphase and cytokinesis. The answer will be N, 1, and 3. Number 20, telomerase is an enzyme that adds nucleotides to telomerase. Which statement about telomerase is correct? So in this case, the answer for this question is D. The low concentration of telomerase in body cells mean that these cells can divide a limited number of times. And telomerase is actually an enzyme that extends the telomerase at the end of chromosome, which helps to maintain chromosome integrity during cell division. Most somatic body cells have low or no telomerase activity, leading to the gradual shortening of telomerase with each cell division, which ultimately limit the number of times these cells can divide. And this is a mechanism that contributes to cellular aging and the half-life limit. In contrast, stem cells and cancer cells often have higher level of telomerase, which is allow them to divide more times without experiencing significant telomerase shortening. And number 21, the photomicrograph shows cells at different stages of mitosis. A student wrote 4 statements about the photomicrograph. 1 is actually represent cells P which show anaphase, and 2 is actually splinter formation is occurring in a cell Q, and 3 is actually the amount of DNA in cell R is the same as in cell T. And number 4, the correct order of the stage is S, R, T, P, and Q. Which statement are correct? In this case, they're referring to cell P show anaphase, and 3 is actually the amount of DNA in cell R is the same as in cell T, and the correct order of the stages is S, R, T, P, and Q. 22. Bacterial cells with DNA containing only the heavy isotope of nitrogen, which is N50, are allowed to reproduce for 3 generations in a culture medium containing the normal isotope of nitrogen, which is N14. Which percentage of the DNA molecule produced containing strand with the heavy isotope of nitrogen? Let's see the next page, the explanation. To understand how the percentage of the DNA molecule containing strand with the heavy isotope of nitrogen changes over generation, we need to consider the mechanism of DNA replication, which is semi-conservative. This means that each new DNA molecule consists of one old parental strand and one new daughter strand. Let's go through the generation step by step. For the first generation, during the first replication, each 15N label DNA strand serves as a template for the synthesis of a new 14N label strand. And each resulting DNA molecule will have one and 15N strand and one 14N strand. And therefore, 100% of the DNA molecule will contain one heavy strand and one light strand. And for the second generation, each of the DNA molecule from the first generation replicates, and half of the daughter molecule will inherit a 15N strand and a newly synthesis 14N strand hybrid, and the other half will compose entirely of 14N strand, and thus 50% of the DNA molecule will still contain one heavy strand. And for third generation, the hybrid DNA molecule, one 15N and one 14N strand, will produce one hybrid molecule and one 14N only molecule. And the 14N only molecules will produce more 14N only molecules. And consequently, 25% of the DNA molecule will contain only heavy strand. So based on this analysis, the correct percentage of the DNA molecule containing strand with the heavy isotope of nitrogen are: first generation is 100%, second generation are 50%, and the third generation is 25%. So the correct answer will be C. A bacteria's circular DNA molecule is 2,600 150 base pair long, and 26% of the base are adenine. And how many cytosine bases would be in the DNA molecule? To determine the number of cytosine bases in a bacterial circular DNA molecule, we can follow these steps. First, we have to calculate the total number of adenine A bases, and given that 26% of the bases are adenine, so the number of bases is equal to 0.26 multiplied by 2,600,150, and you get the value of 6676039. And using Scarver rule, and in the double-stranded DNA, the amount of adenine is equal to the amount of thymine, and the amount of cytosine is equal to the amount of guanine. Hence, number of T bases equals to number of A bases, which is 676,039. Number of T bases equals to the number of A bases. So the value will be 676,439. Calculate the combined number of adenine and thymine bases. Total number of A and T bases is equivalent to 676,039 plus 676,039. You get the value of 135,2078. So the total number of A and T bases is 676,039 plus 676,039. You get the value of 135,2078. And number 6, you have to calculate the remaining bases, which are cytosine and guanine. And the total number of C and bases is equivalent to 26,00150 minus 135,2078, and you get the value of 1248072. So the answer, and then since the number of C bases equal the number of G bases, so the number of C bases would be 1248072 must be divided by 2. And then you get the value of 624036. Therefore, the number of cytosine base in the DNA molecule is 624036. The correct answer will be A. 24. Which statement relating to structure of DNA is correct? The correct statement relating to the structure of DNA is A, which is the alignment of bases to form a double helix is only achieved between anti-parallel strands. And A is not correct because the two DNA strands are joined to each other by hydrogen bond between complementary bases and not by phosphodiester bond. Phosphodiester bond link the sugar and phosphate group within each single strand. B is actually correct, which is in DNA, the two strands are anti-parallel, meaning that they run in opposite direction. One strand runs 5 to 3 and the other runs 3 to 5. This anti-parallel arrangement is necessary for the bases to align properly and form hydrogen bond, which is resulting in a double helix structure. So the answer C is incorrect because 3 hydrogen bonds are formed specifically between the base pair guanine and cytosine and not between all base pair containing purines. Adenine pair with thymine via 2 hydrogen bonds. And D is also incorrect because the number of cytosine bases always equal to the number of guanine bases, and the number of adenine A bases always equal to the number of thymine bases in a DNA molecule, not the number of cytosine bases equaling thymine bases. Number 25, a student sketches a diagram to represent the process of transcription, and which part of their diagram show the non-transcribed strand? The answer given here is A. Number 26, which row is correct for the movement of water in the root? The correct answer is actually A. Why? The apoplast pathway, water moves through the cells, wall, and intercellular spaces, and the Casparian strip, which contain submarine block, the apoplast pathway, which is forcing water to enter the symphyplast pathway to cross into the vascular tissue. So B is incorrect because the apoplast pathway involves movement through cell wall and intercellular space, not through plasmodesta, and the Casparian strip contains submarine liquid. And number C is also incorrect because the simplest pathway involves water movement through plasmodesta, but the Casparian strip contains submarine liquid. And D is also not correct because the simplest pathway involves movement through plasmodesmata, not intercellular space, and the Casparian strip contains submarine. And number 27, and the table contains some information about uptake and movement of water and the mineral ions in the plant. Using the information provided, which factors will affect the uptake and movement of water of the mineral ion in the plant? Whether humidity, surface area of the root hair cells, oxygen concentration, or temperature. Okay, the answer here is the factor affecting the uptake and movement of water and mineral ion in plants are influenced by various environmental and physiological conditions, and based on the provided table and understanding of the plant physiology above, humidity is also affect the rate of water evaporation from the stomata, which in turn can influence the water uptake by osmosis as the plant tries to maintain water balance. And humidity, which already explained, and then surface area of root hair cells, the larger surface area increases the capacity for water and mineral ion uptake. And for the oxygen concentration, it is necessary for respiration, which provide the energy ATP needed for active transport of mineral iron. And finally, temperature also needed, and very important, which is affect the rate of diffusion, osmosis, and active transport, as well as the overall metabolic activity of the plant. Therefore, all four factors like humidity, surface area of the root cell, of root hair cells, oxygen concentration, and temperature play a vital role in the uptake and movement of water and mineral ions in plant. Thus, the correct answer will be 1, 2, 3, and 4, which is A. 28. Which changes to the water potential and the volume of solution in the volume sieve tube occurs when sucrose is moved from a photosynthesizing into the volume sieve tube. And when sucrose is moved from photosynthetic leaf into the volume sieve tube, the following changes occur. The water potential in the volume sieve tube becomes lower because the addition of sucrose, a solid, decreases the water potential, and the volume of solution in the volume sieve tube increases because water moves into the volume sieve tube by osmosis due to the lower water potential. So the correct answer will be D. 29. A student wrote the following statement about a possible mechanism for loading sucrose from a source, and which statement are correct. To determine which statement are correct about the mechanism for loading sucrose from a source, let's analyze one by one. When energy is releasing from ATP, the released energy is used to load sucrose through a transporter protein in the companion cell membrane. And this is correct because ATP is used to pump proton, H ion, out of the companion cells, creating a proton gradient, and the energy from the gradient is then used by the co-transporter protein to move sucrose into the companion cell along with the proton H positive ion returning to the cell. And as sucrose is moved into a companion cell, the pH in the cell wall of the companion cells decreases is also correct. So, the proton gradient created by the ATP-driven proton pumps result in a high concentration of H positive ion in the cell's wall upper plus leading to a decrease in pH. And number 3 is incorrect because sucrose moves from the companion cells to the volume sieve tube through the plasmodesmata or cystoplasmic connections, not by protein pump. So the correct answer would be 1 and 2, which is B. And number 30, the diagram shows a transverse section through an artery. Which tissue are present in the layer X? The layer X is actually represent elastic fiber, collagens, and smooth muscle. Let's look into number 31. What is systolic blood pressure? Systolic blood pressure is the measurement of the pressure in the artery when the heart, specifically the left ventricle, is contracting and pumping blood out into the body. So, the correct answer is actually systolic blood pressure is the maximum blood pressure in the arteries. The diagram shows pressure changes in the left side of the heart and aorta over time. The length of these cardiac cycles is 0.6 seconds. Points 1, 2, 3, and 4 indicate when intraventricular valves and semilunar valve either open or close. What is the total time during one cardiac cycle that the atrioventricular valve and the semilunar valve are both closed at the same time? The answer will be 0.07. Not even 1 second, not even 0.1, nearly to 0.1. Which reaction takes place in the capillary surrounded and alveolus? So to determine which reaction takes place in the capillary surrounding and alveolus, we need to consider the process involved in gas exchange and carbon dioxide transport. Number one, this reaction typically takes place inside red blood cells and not in the capillary surrounding the alveolus. The enzyme carbonate hydroxy catalyze this reaction. Number two, the process number two, this reaction does occur as hemoglobin bind to carbon dioxide to form carbaminohemoglobin. However, this is more relevant in tissue where carbon dioxide is being put up rather than in the lung where carbon dioxide is being released. Number three, this reaction occurs when hemoglobin releases hydrogen ion as it binds to oxygen, and the H-positive ions are then typically used to convert bicarbonate back to carbon dioxide and water. And number four, this reaction is crucial in the lung. Bicarbonate ions are converted back to carbon dioxide and water to be exhaled. And this occurs in the red blood cells surrounding the alveoli. Given this analysis, the correct set of reactions that take place in the capillary surrounding and alveolus is 3 and 4. 34. Which statement explains the importance of the chloride shift in the red blood cells? The correct answer is hydrogen carbonate ion diffused into plasma from RBC and chloride ion diffused into RBC to maintain a balance of positive and negative ion. So the explanation is the chloride shift, also known as hamburger phenomenon, and it is a process that maintains the electrochemical balance between the red blood cell and the blood plasma during the transport of carbon dioxide. Here is how it works. When carbon dioxide enters RBC from the tissue, it reacts with the water to form carbonic acid, which quickly dissociate into bicarbonate ions and hydrogen ion. The bicarbonate ions then diffuse out of the RBC into the plasma. To maintain the electrochemical balance, chloride ions diffuses into the RBC from the plasma to balance the charge. Number 35, and the photomicrograph show a cross-section through the lining of parts of the respiratory system. And which statements about the photomicrograph are correct? So the answer will be goblet cells are visible between squamous epithelial cells and the smooth muscle is visible. The surface tension of the layer of liquid lining, the alveoli tends to pull the wall inward so alveoli to collapse. Which statement would explain how this is prevented? So the answer will be the epithelial cells secrete a chemical that reduces the cohesion in water, which is the surfactant secreted by epithelial cells type 2 alveolar cell reduces the surface tension, thereby preventing alveolar from collapse. What will reduce the rate at which bacteria become resistant to antibiotics? The answer will be prescribing 2 antibiotics with different modes of action. And number 3 is finishing a prescribed course of antibiotic. The answer will be C. And number 38, T lymphocytes have a protein PD-1 on their surface. Some cancer cells have a receptor molecule on their surface which bind with PD-1 in activating the T lymphocyte. A monoclonal antibody, lambrozolzumab, have been produced against this receptor. Trials show that in 54 of 135 people with advanced skin cancer who were given lambrozolzumab, the tumors more than half in volume. In 6 of the 57 people will be given the highest dose to tumor disappear, which may be correctly concluded from this information. The answer will be 1, 2, and 4. 1, 2, and 4, which is 1. This statement is correct because lambrozolzumab MAP is a monoclonal antibody that binds to the PD-1 receptor of its ligand or cancer cells, preventing the inactivations of lymphocytes. And two is actually, this statement is correct because by binding to the PD-1 receptor or its ligand, pembrolizumab prevent the cancer cells from inactivity to lymphocyte, allowing the immune system to attack the cancer cell. And number four, this statement is also correct because by blocking the interactions between PD-1 or T lymphocyte and its glands, ligand on cancer cell, pembrolizumab enhances the ability of the immune system to recognize and destroy cancer cells. Number 39, a person's blood group is determined by antigen, which is present on the red blood cells. And the table shows the antigen and antibodies in the blood of people with different blood group. And during the blood transfusion, it is essential that the person receiving the blood does not have antibody to the donor blood. Which blood group can be given to a person with blood group AB? A person with the blood group AB has both A and B antigens on their red blood cells and does not have antibody to A or B in the plasma. This means that they can receive blood from individual with any blood type, as the immune system will not react against the A or B antigen present in the donor blood. So the answer would be D. Which type of cells are stimulated to divide by the cytokines produced by T cells? The answer would be D, lymphocyte and T killer cells. Why? Because cytokine produced by T helper cells, such as interleukin, play a crucial role in the activation and proliferation of both T lymphocytes, which produce antibodies. And T killer cells, also known as cytotoxic T cells, which destroy infected or cancerous cells. The answer will be D. By leveraging past examination paper, you can enhance your understanding, improve your exam technique, and ultimately achieve better results. Thank you for your attention and best of luck in your studies. And don't forget to subscribe to Zoom Study Learn YouTube channel.